In a previous video, we talked about how the body can create heat, and especially in muscles, by trying to rapidly produce ATP. Rather than creating the maximum amount of ATP, you can rapidly produce it using the glycerol 3-phosphate shuttle, and that actually donates high-energy electrons to coenzyme Q, which means fewer protons are pumped across the shuttle for the same energy, so the rest of the energy is dissipated as heat. There's another way to dissipate energy as heat, and that happens largely in brown adipose tissue. And so what we get is the uncoupling protein. So when you uncouple the flow of electrons from the creation of ATP, so typically flow of electrons is coupled to the creation of ATP. But in brown adipose tissue, there's a protein called uncoupling protein that will allow protons to flow through into the inner mitochondrial matrix without creating ATP. And so that by doing that, you generate a lot of heat because all the extra energy is just dissipated as heat. Now, this uncoupling protein is known as thermogenin, and we're going to take a quick second to see how it works. So what happens is your hypothalamus will sense cold, and it will through sympathetic nervous system release norepinephrine onto the brown adipose tissue, activating adenylate cyclase, creating cyclic AMP, and cyclic AMP will activate protein kinase A. The protein kinase A will activate the triacylglycerol lipase, which basically takes these triglycerides and splits them into 3 fatty acids. 3 free fatty acids for each triglyceride, and the high amount of fatty acids will cause thermogenin to open. Now, typically, di and trinucleotides will keep this closed, but a high concentration of fatty acids will overcome that and open the channel. And in order to keep the electron transport chain pumping protons in, free fatty acids undergo beta oxidation, producing NADH, and the NADH continues to donate electrons, pumping protons through, and then they release all of their energy as heat and come back through the uncoupling protein. Now, the last thing we want to look at in the electron transport chain are things that other things besides uncoupling that can stop the flow of electrons, and that'd be poisons. So respiratory poisons are things such as insecticides, barbiturates, antibiotics, and antifungals. So, for example, you can stop electron flow into complex 1 by retinone, amytal, and mixithiazole, and stop the flow at complex 3 by antimycin A, mixithiazole, and stigmatelin. So you see mixithiazole stops at both. Complex 4 is stopped by cyanide. So that's what cyanide does. It stops electron flow at complex 4. Carbon monoxide and azide will do the same thing. And then you can have things like uncouplers that will cause proteins to leak through the inner mitochondrial space without producing ATP. And so just to kind of zoom in, you remember we said that there's 2 subunits. There's an F0 and an F1 subunit for ATP synthase. The F0 subunit is basically what allows the flow of electrons through, and the F1 subunit is has the catalytic activity that turns ADP and inorganic phosphate into ATP. And anything that would inhibit either of those activities would cause electron leakage or proton leakage. And specifically, since we're talking about the synthase, we'd be talking about proton leakage.